Okay, ladies and gentlemen, you are welcome. So in today's video, we are going to start with retrieval of gene sequence. So how do you retrieve gene sequences from different databases? And don't forget, from my previous video, I discussed about the different types of databases and their applications. So why do we retrieve genes? We retrieve genes for so many uh, applications. We can retrieve genes to design a primer. So you can decide to retrieve your gene of interest. You can obtain your gene of interest from different databases in order to design a primer for GCR. And also at the same time, you can retrieve a gene sequences in order to generate a phylogenetic tree to compare the genetic or the evolutionary relationships between different species of organisms. You can use that at the same time. So now, ladies and gentlemen, in this video, I'm going to show you how to retrieve gene sequences, and also I will also show you how to store gene sequences, including the address of the genes. So let me share my browser. So this is my browser, my Chrome browser. So the first thing to do here, I have my browser here. So let's start with the NCBI. Let me use NCBI. NCBI is stand for National Center for Biotechnology Information. It is a house, or it is a National Institute of Health house that actually houses different databases. So if we click on it now, it will take you to the page where we have all the databases. As you can see, all these are databases where you have assembly, we have bio collections, bio project, bio sample books, Climber. We have many of them. We have genes, genome, CEB data sets. We have many of them. So if you are looking for a gene, you can click on either this gene database or nucleotide database. So, but at this point, I'm going to use a nucleotide database. So when you click on it, let's say you already have your gene of interest, that is the gene that you want, or that you want to get the sequence of of your gene of interest. So you write it here. So let's say I'm looking for, let's say, hexokinase in microsporidia. Let's say hexokinase in microsporidia. So let's search. So you see, these are the different genes. So the different hexokinase genes from microsporidium. So this is microsporidium, Daphne, hexokinase. So we have different species of microsporidia. So it depends on the species of the microsporidia that you are looking for. We have microsporidia MBSA species. We have Nosemia. We have Nosemia tyria. We have different species of the microsporidia. So let's see. Look at them here. We have different of them. So this is Encephalitozoan corniculi. It is one of the species of the microsporidia that causes disease in humans. And the name of the disease is microsporidiosis. So let's say now you are interested to develop a drugs. You are interested to develop a drugs against these species of microsporidia. That is encephalitozoan caniculi. Or maybe you want to identify it in a blood sample. Let's say you collected a blood sample from a particular individual and you want to see whether that individual is harboring this species of the microsporidia. So how do you do it? Maybe you wanted, or you are interested to get the primer. So for you to get the primer, you need to get the gene first, and then you use it to design a primer. So let's click here. So when we click here, when we click on the gene, so it is going to take us to this page. So generally, if you want to retrieve your gene of interest, you usually get it in FASTA format. So this is a FASTA. So you click on FASTA. So you see it is now taking us to the page. So you see these are the number of nucleotide sequence. As you can see from here is 1540. And this is what you call assertion number. This is assertion number. It is like a ID. It is an ID of each and every gene. So if you can even take only this assertion number, it will always, you can use it to search for your genes in any database and it will give you everything. So this is the assertion number. Instead of writing the name of the genes and the species, this one is an ID that will give you exactly the species of the species and the gene that you are specifically looking for. So now from here, we now have our genes in the FASTA format. So we copy it. So we're now going to copy it. And then you are now going to your Microsoft Word. Okay, I already have this. So you can now create new. So you can paste it here. So this is your gene sequence. So you can leave it here. If you want to take any other one, you can also copy it and paste it here. Maybe you want to do sequence alignment, and maybe in human we have hexokinase, and also we have it in the encephalitozoan caniculi, and it is a parasite of humans. So you know that you cannot develop a drugs. You cannot develop a drugs that actually the organism have the same, or the two organisms, both the parasite and the host, are having the same gene, and also in terms of the similarity, the level of similarity is very high. So you can't do that. So if you want to check the level of the similarity, like this is Microsporia species, so you can also get the hexokinase from humans, and we also paste it here. After you do that, then you copy the sequence and check the percentage similarity. So this is how we retrieve genes. You opened a Word and you name the Word document, and then you paste it. And after you paste it, then you can now leave it for other downstream application. So ladies and gentlemen, thank you very much. This is actually how to retrieve gene sequences, and we are going to continue in the next video. So we are now going to see how we can use this sequence of encephalitozoan caniculi, design a framework for both conventional PCR and qPCR. So follow me in the next video.